Hey, good morning. Thank you to FreeMind for recognizing Sapience as one of the elite companies that they work with and for the opportunity to speak to everybody here today. So our quick disclaimer page. Sapience Therapeutics was founded in 2015 with the idea of targeting hard or difficult to treat cancers by going after what were previously considered undruggable targets. And in order to do this, what we did was design a new therapeutic modality we call SPEARs. SPEARs are stabilized peptides that are able to identify and bind to a target, a previously undruggable target, disrupt its interactions with other essential cofactors, and thereby prevent its activity. We've designed SPEARs against the transcription factor CBP beta. That is our lead clinical SPEAR and the prototype of this modality. ST-101 is currently in a clinical study evaluating its activity in patients with glioblastoma. We've seen several successes thus far in the clinic and have ongoing studies that I'll talk about in just a few moments. Our second clinical SPEAR is ST-316. This is designed against pretty much the quintessential undruggable target and the reason for companies such as Sapience are in existence, and that's beta-catenin. We have devised and designed this peptide to attack the oncogenic activity of beta-catenin while leaving its homeostatic functions fully capable. What this enables us to do is separate or bifurcate the oncogenic versus the pathologic functions of beta-catenin and deliver a completely safe molecule, whereas enabling us to attack this very difficult and elusive target. The ST-316 program has recently entered a phase 1 clinical study in a bucket of patients who are likely to harbor Wnt mutations, and we expect to start a phase 2 dose expansion study later this year in patients with colorectal cancer. Our SPEARs approach and the design of these molecules has enabled us to identify that we could do this against other previously undruggable targets as well, and that has enabled us to design our pipeline and our platform discovery system, which we term ADAPT. ADAPT enables us to screen multi-million peptide libraries against full-length proteins in a cellular environment, enabling us to identify and isolate disruptors of those protein-protein interactions for further optimization and ultimate clinical activity. We have thus far received NIH support in the form of SBIRs to support both the ST-101 and ST-316 programs, as well as some of our developmental programs to the tune of about $4.6 million. It's been a very successful program. It's enabled us to really expand our research and enable us to characterize these molecules in a way and with outsourced activities that maybe would be difficult to do under normal and current budget situations. So it's been a very successful system, and I would echo previous comments that say that it really has not in any way, shape, or form distracted us from our goals and our mandates. We've designed and written these grants with the intent of following the path that we would probably be following anyhow, just enabling us to expand to a greater degree, and that's really been a very good formula for us thus far. So I mentioned a new therapeutic modality called SPEARs. SPEARs are stabilized peptides, and they are designed against protein-protein interactions. Why are SPEARs necessary? Well, our current, biotech industry in general looks at protein-protein interactions as undruggable because whereas small molecules are really adept at getting into cells, reaching protein targets, they require a kinase activity or a binding pocket to exert their activity, and most protein-protein interactions just simply don't have that. Antibodies are excellent at blocking two proteins from coming together, but for the most part, antibodies can't enter the cell, enter the nucleus, where these protein-protein interactions are really essential. So our SPEARs, our stabilized peptides, are able to get into cells, they're able to bind a target, we can send them to the nucleus, we can send them to the cytoplasm, and they can exert their activity. They block binding interactions that are essential for two proteins to either sit on DNA or exert another signaling event, and that is really the benefit. Because of the process in which we stabilize our peptides, we use D-amino acids, versions of natural amino acids. These peptides are not liable to proteolytic degradation. As a result, they are not processed by MHC, and they processed and presented by MHC, and therefore they're non-immunogenic. We have seen plasma half-lives in the range of 2 days, whereas a standard peptide that you might consider an L-amino acid peptide would have a half-life of minutes to at most hours. And they are exceedingly well tolerated because they are targeting specific protein-protein interactions that are required for oncogenic activity, however, that are not essential for normal everyday functions. So over the next couple slides, I'll give you a brief introduction of the ST-101 program and some of the clinical data that we've generated to date, and as a demonstration of our SPEAR platform. ST-101 targets the transcription factor CEBP beta, not exactly a household name under most transcription factors. However, a really interesting and important target in that it has both direct oncogenic activity within tumor cells, as well as an important effect in the immune microenvironment. CEBP beta is expressed in almost all cells. However, in most differentiated adult epithelial and endothelial cells, it's held in a repressed state. When it is active, and that's primarily in adults and in tumor cells, it activates a transcriptional program that enhances survivin, IDS, cyclins, essentially embryonic stem cell factors that enable tumors to proliferate, de-differentiate, and show chemo-resistant activity against most standard drugs. In addition, within the tumor microenvironment, CEBP beta is expressed in a subset of cells called M2 macrophage. These are immune suppressive macrophage that act by making what we term the tumor microenvironment cold, excluding cytolytic T cells, enhancing the recruitment of regulatory T cells that further block the immune response. In the presence of ST-101, CEBP beta is not able to dimerize, and by disabling its dimerization, it's not able to sit on DNA, and it's not able to enact its transcriptional program. In the tumor, we see direct anti-tumor activity by tumor cell cytotoxic activity by decreasing all these survival factors that are essential for the tumor growth. But what we also see is a remodeling of the tumor microenvironment. We see the shift from M2 macrophage to the immune supportive and pro-inflammatory M1 macrophage, therefore seeing recruitment of CD8 cells and direct anti-tumor immune responses in addition to the direct anti-tumor cytotoxic events. As I mentioned, ST-101 is currently in the clinic. We are completing a phase 2 study with the intent of starting a phase 2b later this year in glioblastoma. This is a demonstration, and I hope some of you can see some of the immunohistochemistry that we have up here. But this is a direct demonstration of both ST-101 passing past the blood-brain barrier into the glioblastoma tumor, and that's depicted on the top. This is immunohistochemistry to ST-101 itself. And post-treatment, you'll see a brown stain throughout the tumor, as well as target engagement. On the bottom, we see CEBP beta immunostaining within the tumor is decreased post-treatment. This is a demonstration and supports what we've observed in vitro and cell culture studies, showing that direct targeting of CEBP beta promotes its proteolytic degradation by the proteasome. So we see both tumor uptake and target engagement. And here's a case study of a patient who's a 34-year-old male, came into the clinic with glioblastoma and was recurrent glioblastoma, and was put into our ST101 phase 2 program. This patient had a tumor that we can see at baseline was actually pushing over the midline and was considered a very poor prognosis. Over the course of several months of treatment, and this patient remained on study for over a year, we saw the tumor shrink continuously to a 93% loss of tumor. This was considered a partial response, not a complete remission, because we were not able to biopsy the tumor and demonstrate that there was no tumor cells left. However, this patient has received extended life, essentially, in a disease that is very difficult to control. We are additionally seeing some clinical responses in our phase 2 study, both partial responses and in a window of opportunity sub-study in which drug was given before and after tumor resection. We see additional responses, including a complete response. So we're very excited by this program. We're very excited by the prospect of using our spears for difficult to treat cancers. Here's just a summary of where our company currently is in terms of clinical evaluations. The ST101 and ST316 programs are progressing nicely, and our next programs, our discovery programs, are essentially ready for IND-enabling studies. We have a program that targets the AP1 complex, specifically the Jun-Fra1 interactions, which are essential for many gynecologic and similar cancers. In addition to multiple 7 discovery programs that are in different stages of hit to target optimization. So with that, I say thank you. I appreciate your time and interest, and I'm happy to answer any questions you may have, both on our funding path as well as our programs. Yes? The second lesion started while the patient remained on study. So with the occurrence of a new lesion, they come off study. We are currently trying to sequence that tumor and identify if there were specific mutations that would teach us, patient selection for future studies.
It's still very interesting. It's really exciting.
I appreciate that. Okay, well, thank you very much.